7.1, which is an overview lesson going from DNA to RNA to the completed protein. There's two major parts to it, so I'll just flip to the next screen so you can see it in picture form. So here I have the DNA. It's double-stranded. We undergo a process called transcription, where we get a single-stranded mRNA. It doesn't need to be double-stranded. It's going to act like a template. This goes out to the cytoplasm and is read or translated into this polypeptide strand, which is amino acids linked together. After that, everything detaches, and then some post-translation things happen to make the full protein. Okay, so we can see we have transcription, where DNA is copied to RNA, and then translation, where amino acids are fetched from the cytoplasm and linked together. So DNA is single-stranded. DNA, this is double-stranded. RNA is single-stranded. Another difference is that instead of thymine, we have uracil. A third difference is that this is the sugar that's used in DNA, deoxyribose sugar, which means it has one less oxygen, which is at the number 2 carbon. This is ribose sugar, which is used in RNA. You can see that there's an oxygen here. So here is just a list of the comparisons. So RNA is single-stranded, whereas DNA is double-stranded. We have the ribose sugar here. DNA has deoxyribose. The DNA can't leave the nucleus because it's a blueprint, so it can't get damaged or whatever. The RNA must leave the DNA. The RNA must leave the nucleus in order to get to the ribosome where protein assembly happens. So this is just a one at a glance about the differences. So once again, we have the OH here instead of the H. We have single-stranded RNA instead of double-stranded. These are the same, but here we have the U instead of the T. Okay, so a new player here is tRNA. So you can see its characteristic cloverleaf shape, and its job is to go into the cytoplasm, fetch amino acids, and bring them to the ribosome to link together into a polypeptide. So there's different types of RNA. There's mRNA, tRNA, and rRNA. So the mRNA is the one that I showed you before. You have DNA being transcribed into RNA, which leaves the nucleus. Then there's the tRNA, which is sort of the fetch and carry tRNA, which goes and reads the triplet codon at the mRNA at the ribosome, and then goes and finds the correct amino acid and brings it back. And then this is within the ribosome. Okay, once again, we're going to see this again and again, but you have your DNA template. Transcription gives you mRNA, and translation gives you a protein. So all this overall is called the central dogma. Okay, so first of all, I'll come back to that in a minute. So once again, we have the DNA that forms the template strand. So the DNA has been unwound. You get a single strand of it. And then over here, you can see this mRNA. So at a glance, the way that you can tell that it's mRNA is that it has U instead of T. So if you have A, you would usually have a T, but now we have a U. So that's what happens when you get DNA becoming mRNA. Then the mRNA goes to the ribosome and is translated based on these three letters or bases. So these three letters, these specific letters, code for a certain amino acid. These other letters code for a different one. These other three code for this one. This other three codes for this one. You're never going to have to memorize this. You'll have a chart to use. Okay, so we go back. This is just sort of saying what I already said. This is already what I said. Okay, so once again, there's a lot of repetition in this lesson, but hopefully you get it. You have your double-stranded DNA. It is transcribed to give you pre-mRNA. A few things happen in order to get mRNA. So such as these non-coding bits are taken out, and there's a cap and a tail to protect it from being degraded in the cytoplasm. Then the mRNA is able to go through this pore, through the nuclear envelope or membrane. It combines, you can see the mRNA here, with this hamburger-looking thing, which is the ribosome, and then translation happens. So here is the codon chart. You will always be given this, and we use this to figure out which amino acids are coded for. So for example, if you had a U, and then something else, so UUU or UUC, UUA, UUG, these code for phenylalanine or leucine. There's other things here too, which are things like stop and start codons. So you can see this is the start codon in green, and these three are the stop codons. More on that later. Okay, so on the RNA, you have combinations of these three. So you could have AAA or UUU or AU or whatever, and it codes for something. There are 20 amino acids. This 3-letter code is called a codon, and it's a start codon that codes for methionine. And that tells the cell that that's the start of a polypeptide chain or a chain of amino acids. And then, as I said before, these three are the stop codons. And once the stop codon is reached, then the ribosome and the mRNA goes its own way. There's something called a wobble hypothesis, and that accounts for any mistakes that are starting to happen. So it results in a redundancy. So if you have CCU or CCC or CCA, CCU, they're all code for the same thing, that's proline. So if there's an error at the last base, then you'll still get the same protein, which is really great. So you can see some amino acids and their abbreviations here.